Hello, everyone. My name is Yaron, and I'm a graduate student at the Chemistry and Elemental Laboratories at Weill Cornell Medicine. First, I would like to thank the organizer for the opportunity to speak here today, and I would like to tell you a story about how we combine biochemistry, computational biology, and virology in order to decipher an interesting mechanism in SARS-CoV-2 infection. So for those of you who knows our lab, we mainly deal with cancer, protein kinases, PI3 kinase, and cancer metabolism. And therefore, for the first part of my talk, I want to introduce you to a system that we have been developing in the past 4 years, and then to show you how we apply that for studying COVID-19. The system is called the Kinase Library and is based on two central assumptions regarding protein kinases. The first assumption is that protein kinases are selective for the central phosphoacceptor, whether it is serine, a threonine, or a tyrosine. But the second assumption, which is the most important assumption for our system, is that protein kinases are selective for the neighboring amino acid sequence around the phosphoacceptor. That means that once a protein kinase will encounter a potential phosphorylation site, it will look at the amino acid sequence around it. The amino acid sequence on the left, denoted in the minus positions, and amino acids on the right, denoted in the plus position. And different protein kinases would like to see different amino acids in different positions in order to carry out the phosphorylation. That favorability of every individual protein kinase is what we call a kinase motif, or the substrate specificity. In order to biochemically characterize the substrate specificity of different protein kinases, we use a technique that was developed in our lab back in 2004 called combinatorial peptide libraries. Essentially, we try to quantify the contribution of every possible amino acid in every possible position to the phosphorylation of the phosphoacceptor. For example, let's say that we try to quantify how a proline in -5 will contribute to the phosphorylation of a central serine or threonine. So we have a peptide which will have a serine or threonine at the center so it can get phosphorylated. All the other amino acids will be completely random, denoted as X, and the Z will be specifically having a proline in the -5 position. And then we have another set of peptides with glycine in -5, and another set of peptides with alanine in -5, and then in all the amino acids in -4, and -3, and -2. And this is why you get a plate of libraries, a plate of different peptides, which all of them will have one fixed amino acid at a time at one fixed position, and all the rest will be completely random. So for example, let's say that we take a look at the -2 arginine well. That will have a bunch of peptides that all of them will be 50-50 serine or threonine in the center, and we have an arginine 2 position to the left because it's a -2 arginine. All the other amino acids, though, will be completely random. And then for every kinase that we try to characterize, we add the protein kinase to the plate with hot ATP, and the more favorable that amino acid is at that particular position, the more phosphorylation will occur. Then we blow this peptide with streptavidin plate, and we quantify the radioactivity using phosphor imager. And the outputs, as you see here, a bunch of matrices, one matrix for a kinase, in which you get dots. Every dot is one amino acid at one particular position type of peptide. And by that, you can see which protein kinases likes better particular amino acid than the others. So how do you read these matrices? All you need to know is the following 5 things. On the left, you see the rows will be the positions. For -5 position on the left, the +4 on the right. This is just how the library of peptides was designed. The columns will be 20 unmodified amino acids plus 2 already phosphorylated residues. So we are able to quantify how an already phosphorylated threonine or tyrosine will contribute to the phosphorylation of a nearby serine or threonine or tyrosine, where phosphothreonine is identical to phosphoserine. And then the dots will be the favorability. The darker the dot is, the more favorable that amino acid is that particular position. For example, for pKa, is really not selective for anything in the -5 and the -4 position, but for the -3 and -2 position, it's highly selective for having an arginine, which is a strong agreement with 4 decades of research of downstream sites of pKa from phosphosite plus. CDK1, on the other hand, is selective for +1 proline and +3 lysine, and all the rest can be completely random, again, in a strong concordance with downstream substrate of CDK1 after decades of research from phosphosite plus. And the last thing that you should note is that the +1 position, for example, for pKa, looks like it's not selective for anything. But if you take a look at the +1 proline, it's much whiter and brighter than any other background noise, suggesting that this is a negative selection. So pKa will probably not phosphorylate any serine or threonine, which is followed by a proline. As you can see here, pKa and CDK1 has completely distinct motifs, and these motifs are totally different for every single protein kinase, of course, more similar within families. So for the entire human kinome, we have about 450 active kinases, the rest are pseudokinases. We've characterized 300 serine or threonine kinases and the entire tyrosine kinome, which can give us a good idea about the entire specificity of the entire kinome, or at least a close family member for every protein kinase from the kinome. So without going into the details of the computational scoring system, because I want to tell the story about the COVID-19, all you should know is that our system takes a substrate as an input, and together with all the motifs that we have in a prediction algorithm, you get an output, which is a sorted list of all the human protein kinases sorted by the favorability score for that particular motif. So what does that have to do with COVID-19? When the pandemic started, we took a look at the virus because we never did virology in our lab, and we saw that it has 4 structural proteins: the spike protein, the membrane protein, the envelope protein, and then we saw it's a nucleocapsid phosphoprotein. Now, we know nothing about viruses, but we know a lot about phosphorylation. We try to understand what is going on there. So we assembled a team in order to ask the 3 questions: the what, how, and why. What is getting phosphorylated? How does that get phosphorylated, by which protein kinases, and why the phosphorylation is important for the viral life cycle. The team consist of cancer metabolism people, mass spectrometry people, and virologists from across the United States. And together we started by a wide characterization of the nucleocapsid protein phosphorylation. After showing you the phosphorylation site, I will show you the phosphorylation model of the nucleocapsid, and eventually we show you how inhibiting these kinases can inhibit viral infection. So first, we just infected A519 viral cells with SARS-CoV-2. We sent them to proteomics and global phosphoproteomics through tandem mass spectrometry. And you can see that particularly the nucleocapsid protein of SARS-CoV-2 is heavily phosphorylated, specifically in a special domain called the SRH domain here in red. Now, comparing the evolutionary conservation of the nucleocapsid protein across multiple coronaviruses from multiple different species, about 82 different viruses, you can see that the SRH domain is as conserved as the NTD and the CTD, which are known to be functional domains, but it's much more conserved than the very adjacent linker domain here, suggesting that it's not only important as a linker, but particularly that domain might be important because it's getting phosphorylated. Diving into that particular domain, you will see that the most conserved amino acids would be either serine or threonine in red or arginine in purple, and all the other amino acids in gray are significantly less conserved than average across multiple coronaviruses, suggesting that the important amino acids in that domain are the ones that are getting phosphorylated or the arginines. So by now we understood that this domain is important because it's conserved across coronaviruses and it's heavily phosphorylated with 14 different phosphorylation sites. And then the question is, what upstream kinases phosphorylate these particular sites? This is where the Kinase Library comes into place. So when plugging in the sequence into the Kinase Library, we saw that there are 2 particular sites, serine 188 and serine 206, with scores particularly high for the SRPK kinase families. And why is that? You will see by your own eyes using just the matrices. The serine 206 here, as you can see, is being surrounded by a +1 protein minus and +3 arginines, which are the darkest spots of the SRPK 1 and 2 motifs, suggesting that these are the most favorable amino acids for this kinase family. The serine 188, though, is being surrounded by +N -2 serines and +N -3 arginines, again, the darkest spot in these kinase motifs. And this is why the SRPK family scores high for these 2 particular cases. Now you will find an interesting phenomenon in which every single site is being followed by a consecutive chain of serine or threonines exactly 4 amino acids apart. And this is interesting because it's related to an interesting phenomenon called phospho-priming, in which an already phosphorylated residue will trigger the phosphorylation of a nearby serine, threonine, or tyrosine. And the most known example is GSK3. As you can see here also by the matrices, GSK3 will phosphorylate only serine or threonines that will have an already phosphorylated serine or threonine for position to the right. So what happens is that once the serine 188 and 206 gets phosphorylated by SRPK, GSK3 alpha or beta will carry out the phosphorylation of this chain every 4 amino acids apart. And the last but not least site is the serine 205. Plugging this site into the kinase library will yield the casein kinase 1 family because it's had a -3 already phosphorylated serine from the first chain and a -4 unphosphorylated serine, which again are the darkest spots in the casein kinase 1 family, kinase motif. Once that gets phosphorylated, GSK3 will complete the phosphorylation of the 3rd chain. So just to conclude, to summarize the phosphorylation model, we predict that the 2 sites would be phosphorylated by SRPK 188 and 206. One that's getting phosphorylated, GSK3 alpha and beta, we carry out the phosphorylation every 4 amino acids apart, will amplify these 2 signals, and eventually casein kinase 1 will complete the phosphorylation of the 3rd chain. And this is how you get a heavily phosphorylated chain of 14 amino acids in the nucleocapsid domain. So according to that model, if we trim the head of the snake, if we To inhibit the SRPK family, this entire cascade should not happen and the virus should be compromised. And indeed, without going into too many details, I'll just show you that after using molecular inhibitors of SRPK1 and 2 that indeed worked, we tried an FDA-approved drug called alectinib, which is an ALK tyrosine kinase inhibitor, what was shown in 2019 to inhibit also SRPK1 and 2. And you can see here administrating alectinib to either cell lines or primary AT2 cells significantly inhibit the virus. Now, I told you that this domain is heavily conserved across multiple coronaviruses, so it might be important for other coronaviruses, since indeed the common cold that we catch every year, the HCOV-229E, is also being inhibited dramatically by low dose of alectinib, suggesting this is a general mechanism for coronaviruses. So just to summarize what I've been showing you today, I hope that you saw that the nucleocapsid phosphorylation is conserved and probably essential for the viral life cycle. I showed the phosphorylation model, which was predicted computationally, but then was proved, or evidences were provided biochemically and biologically, starting with priming SRPK and then GSK3, and then that primes casein kinase 1, and eventually that inhibiting SRPK at the head of the snake will compromise viral infection. And the last question that I want to leave you with is why? Why is this phosphorylation is so important? And without going into details, I would just tell you that we see an interesting phenomenon that when we compare the transcriptomics and the proteomics with and without alectinib and infection, we see that upon drug, we don't see an enrichment of immune pathways in the transcriptomics level, but we see an enrichment of the immune pathway in the proteomics level, suggesting that somehow this phosphorylation has something to do with the translation of immune genes. But again, we are looking into that right now, probably have more data soon. So with that, I would like to acknowledge a big team of all across the world, from Weill Cornell, from Duke University, CST, Mount Sinai, and the Broad Institute for the help in being part of the team. I would like to thank you, and I'm happy to take any questions. Thank you very much.